Today I will present a work in progress for a sound installation called Mycelium Garden. This is a project funded by EUR Artec and MSH Paris Nord. The mycelium is a vegetative underground organism whose fruiting bodies are the fungi. In the case of mycorrhizal fungi, the network can then connect several root systems of sometimes different plants. A symbiosis is observed. There is a sustainable coexistence between two organisms whose interactions are mutually beneficial. In this type of relationship, the plant brings the product of photosynthesis, which is sugar, to the fungus, and the fungus gives, in return, water and mineral elements. Since 1873, we know that plant cells conduct electrical signals. Burden-Sanderson was the first to identify the action potential in plants. And more recently, scientists have revealed action potential in fungi, which is not a plant, it is to be said, and mycelium networks. In 1995, Olson and Hansen measured electrical signals in the mycelium of Neurospora crassa. Deshibi and Adamatsky discovered by a multi-channel analysis the complexity of signal transmission systems in the network of mycelium of Pleurotus ostreatus. This extreme diversity, they say, could then be compared to the complexity of human languages, even surpassing it in some cases. Mycelium Garden is a research creation project that aims to make audible and sensitive the parts of the living which seem inert to the majority of us, in order to reveal the existence and the complexity of non-human life, underground life also. We search for a model of interaction that facilitates human sensitivity to other, a model that is not seeking to induce anthropomorphic empathy reaction, but which tries to keep the dimension of otherness in the non-verbal sensory interaction. We explore the type of mediation which passes by the experiential in order to facilitate a certain attention to the living, an attention that passes by a listening experience through our ears but also to our bodies. The core of the project is the creation of an interactive sound installation in co-creation with a network of mycelium whose emergence, development and living conditions have been organized by humans. We started by observing the mushrooms present in the MSH Paris Nord garden, where our laboratory is, and we found an agaricus which seemed to be a good candidate for cloning. Stephen Whitmarsh, a neuroscientific and musician from our team, started the cloning, transfers, and culture in petri dishes. Spora Collective then continued the division and multiplication of the mycelium. We named this mycelium Ortus anae while waiting for the fruiting phase. Once we have a fruiting body, we will be able to make a DNA test and determine precisely its species. We realized that Agaricus ortus anae is a very fragile mycelium with slow development. Several months were necessary for Spora to understand and adapt to the needs of this organism. There is little to no standard methodology to measure the signal of mycelium. There is no standard of representation for this kind of data. This data can vary greatly depending on the type of mycelium, the substrate on which it is grown, the type of electrode used, and the environment in which the measurement is deployed. As a consequence, there is also no standard approach to model this signal by machine learning. All these parameters motivated the adoption of an experimental method built on trial and error. In this first phase of the project, we decided to make with. This means we used the technologies which were available to us. In the framework of the INR project BBDMI, researchers from the CICM and ICM developed prototypes in 2022 to measure electrical signals from the muscle and brain using electrodes, amplifiers, and analog to digital converters to make music with this signal. In our project, we tested how we can build on this modular technology to the present installation in order to monitor the action potential of the mycelium. In our recordings, we noticed a signal a bit similar to the one measured by Professor Adamatsky's team, but with the absence of the big voltage spikes. We will continue our experiment to determine if these differences in potential originate from the mycelium. The figure shows the signal of the agaricus mycelium still in development in a petri dish. We will continue in the next phases to monitor on a more developed mycelium as well as during the fruiting phase. In order to access to the very slow and low electrical activity of the mycelium, we partnered with Hugo Scurto, a machine learning engineer and a musician. The objective of their work was to develop a form of automatic listening to the mycelium through the electricity it produces. To do so, they proposed to create a machine learning prototype model of the electrical signal measured with the EV board. They employed a diffractive method. Technically, it is about working with small data sets. It was also proposed to use a shallow learning model to model this small data while guaranteeing a real-time use on the CPU. The model detects and classifies these components through the wavelength analyzed data stream of the mycelium electrical signal. The final patch, Max patch, is implemented in Max. It uses IRCAM libraries, Mubu, Pipo, and XMM. Oops. Normally the video starts. It doesn't. It's okay. So you could see on this patch you can load the recording, choose the frequency range, record 2 examples in which the model will search for the class noise and the class mycelium, then train. Then the patch classifies the probability of the mycelium signals to be present or not. The installation is a patching of several technology, human expertise, and living mycelium. Then I asked myself, because I'm the composer on the project, how to connect the mycelium signal to our human musical world. What sensitive mediation and compositional choices can we propose? In the first phase, in this first phase of research creation, we used the recording of the noise, which is the mycelium and its environment, as material. From an FFT analysis performed in SPARE, I looked for frequency singularities within the noise. I was able to identify rare but remarkable intensities in the frequency bands below 50 Hz. From this analysis, I developed a sound poetic inspired by the mycelium and its environment, the underground. Then this material was passed through sound synthesized in Max using the BBDMI and ABClib libraries. The control of this synthesis and the activation of certain sound samples are modulated in real time by the mycelium electrical activity. It is a sort of music mixed between a human composition and the mycelium. The mycelium modulates in real time the parameter of the sound transformation. The work on low frequencies is also part of the hypothesis of establishing a contact with the mycelium. The diffusion of the soundtrack I compose and modulating by the mycelium is done on a hybrid wave field synthesis ambisonic system reinforced by 4 subwoofers. We are working here on the possibility of a reaction of this organism to the vibration of the low frequency, but it is still to be tested. So here, I hope it's working. It's an excerpt of the music filmed during the assembly of the installation. When the mycelium activity goes over a certain threshold, it activates samples that are also transformed in real time by the continuous electrical activity. So, the objects and the installation developed in this project are built as spaces of reflection to rethink the question of interspecies communication, conceived not as a process based on a common culture and intentionality, but as a form of co-production of meaning by adjustment of differences using Dominique Guyot's concept. The setup also invites us to question the aesthetics states of form generating automatically, or in this case, guided by non-human processes, by showing the fragmentation and dissolution of intentionality in this type of production, which synthesize and explore unexplored niches of our cultural spaces. It has been a very humbling process learning to know the mycelium, to keep it alive, and trying to approach its signals and interact with it through music. In the next and second phase of this project, we will test a new recording protocol with equipment that will allow us to access the signal more easily. In the second phase, we will try to build an AI model that will allow us to classify the different types of mycelium, behaviors, and its reaction to stimuli. This will allow us to refine the relationship we are building with this mycelium. By integrating AI development in this context, we will also try to compare intelligences. What do we mean when we talk about intelligence? What would be the intelligence or adaptability of the mycelium compared to that of the human musician and that of artificial intelligence? What do we mean by this? Thank you. So the flickering noises you heard are activated by the modulation of the electrical signals.
Thank you. Very, very interesting project. So the composition came out of doing a fast Fourier transform on the, let's say, the complex waves produced by the the mushroom. How much aesthetic application did you have between getting the data and creating a musical composition? In other words, going from noise to signal.
Yes. So I used a software called Spare, and I used a sort of poetic of excavating the very low frequencies. So I erased all the frequency bands and I stopped at 50 hertz. And then I kind of graphically reorganize it. And then it's also all metaphorical and artistical. So it was then the composer using noises. It is to be said that we know that the signal of mycelium is in there, but there is also electrical activities around. So for the compositional process, it was the whole noise that was taken in account.
Great. Thank you very much.
Merci beaucoup. On peut applaudir évidemment Diane Schuh. Merci.